Good morning, everyone. I am Lokman Sueto. I'm a PhD candidate at the Southern Cross Plant Science. I will present the ICGRC web portal. The ICGRC, or International Cannabis Genomics Research Consortium, was formed out of group meetings from PAG since 2018. And in 2020, Prof. King suggested that we develop a web portal to host publicly available cannabis research data. And here I'll describe what we have done. So the Cannabis ICGRC site, this is the front page. The site is based on the Tripal framework or Tripal platform. We use Tripal because first, it's part of the GMOD groups. So many bioinformatics and plant or genomics databases already use it. So there's a set of modules that we can readily implement. And then also Tripal has the feature for personalization, meaning a user can log in and given permissions, and he can view only data that he has permission. There's for the same site, there's a publicly available that can be viewed in the internet, and it can also be used as a community platform where individual members can sign in and share group specific data. And then there's a curator who has control, can edit the contents of the pages and the data, and there's an administrator who manages the site. So that's the main reason why we use Drupal. So the links in the front page are shortcuts to specific pages within the site. So here we introduce what's new, what tools are there, and what new data are available. So this data, for example, we generated variant calls from publicly available NGS sequences, and we align it on CS10, Purple Kush, and Phenola, and made that data available here. Okay, so in the menu, there's the ICGRC, introduction to the ICGRC. What are its goals? The aim basically is, since lately there's a change in the legal framework for cannabis, there are legislations that now allow groups to perform research, particularly on genomics, genetics, and phenotyping. And ICGRC collect those reviewed, published research and load it in a portal. So the aim here is to share data and to provide or establish standards that the community can use. And standards can include how genes are named, how the chromosome is to be named, how the cultivars are named. So these things, which is common in any genomics research. And then information on the site. And this is only a part of a system of software. So this is the front page. We're also developing a backend which consists of plant genomics analysis using the Galaxy workflows. And then another is a set of omics analysis tools using statistics and latest web technologies, and probably an API that can serve this multi-omics data. That's the overview of where this project belongs. Okay, in the Infome menu, we start with the genome assemblies. So the genome assemblies are the assemblies loaded to the system. We loaded Phenola, Purple Kush, and CS10. And then there's also C. sativa, which is used for placeholder for others or any cannabis not related to these three. And then Data standards is where we place our agreed standards. As I mentioned, we have many items to standardize, and we will place the results of the discussions here. Then data submission, we are using Drupal, and Drupal is already used in many plant genome research community. We are using their templates to submit. So for bulk loading, the researcher have to contact us, or we look for new publications that have data worth loading, and we format that data into the templates, and the administrator will load those data by bulk. It is also possible to submit data one at a time for curators, but this is for bulk upload. And then there's a user's manual page, which basically describes how to use the site, which I will introduce also today, and tutorial videos on detail for each feature. And then you can contact us, the administrator or ICGRC steering committee, to this page. We also created special interest pages. So since we are a very specific community, we have specific interests. For example, we have those interested in cannabinoid pathways or in terpenoid pathways. So I created just a sample page where it lists the genes and molecules related to those pathways and the links. And this is community driven. So anyone who has a suggestion what to put here, or even curators can edit this page, and we can add different interest links as necessary. So that's why we are using this platform for it to be editable by certain individuals. Okay, I will now show the different features that we developed. The genomics menu refers to the genome and sequence related features. So let's start with the genome browser. We use JBrowse to show the different genome features. So these are the 3 assemblies that are available. And in each page, there's also a detailed description on the sources of data that we use and the method that we implemented. Most cases, there are no complex operations or long workflows. These are just loading, download from source, then transformation, and loading to the database. Okay, so sample the CS10 JBrowse. I know that this is a popular tool for genome research. So the features are grouped into genes, transcripts, and there's also transcript that we generated ourselves, and variants that we also generated using standard pipelines. Okay, so for example, for CS10, this is the RefSeq annotation, and there you can also have the ensemble annotation. Then the transcripts, we downloaded the available transcripts in the public domain, which is Phenola Kansatri or the Purple Kush and CanBio 2. We align it with BAM and load the BAM to JBrowse. And then also the Phylos NGS data in CBI, we downloaded and ran GATK on it and also loaded the features. So this is the Genome browser for CS10, and the same thing for Purple Kush and Phenola. Okay, then the gene function search. For the gene function search, we query the genome for the genes using certain constraints like chromosome region, the transcript accession ID or gene accession IDs, and keywords using the functional annotations. So the genome assembly, for example, we select CS10 and the annotation for CS10. Then we select the region, somewhere between 1, and then search. So this lists the genes and mRNAs for that region. You can also constrain, for example, using keywords like terpene, and this returns the genes with terpene. If we look one gene, for example, it will show the details of the gene, of the annotation. Okay, we go inside here. So for example, so there's an annotation here, and the gene consists of the transcripts. So it has, these are the mRNAs of the gene. If you look at the mRNA, so it has the sequence and also the relationship. Relationship refers to how the genomic features are related. For example, a gene or an mRNA is part of a gene, and then a protein is derived from this mRNA. So these are the relationship. So for this mRNA, we can go to the protein. The protein page also has its own properties, attributes. So it has annotations. So these annotations were run on Go and Gene Ontology and EnterPro. So we searched for the term terpene, but the original gene annotation doesn't have the word terpene, but the word terpene is found in the protein annotations. So in the gene function search, the gene search will search both the gene, the mRNA, and the protein annotations. So I ran Interpro and Go analysis on the CS10 and also on the PK and Phenola. And as a note, since only CS10 is annotated in NCBI, I ran a Finder annotation pipeline on the Purple Kush and Finola genome. So I use Finder because it's straightforward. It uses a combination of Breaker, Augustus, and Peace class for protein annotation using mRNA-seq data. So it's not manually curated yet, but I already loaded it and annotated with Interpro and Go. Okay, so that's the gene function search. And then the genotype viewer. The genotype viewer, we use available NGS datasets in NCBI. So the sources are listed here. So these include publications from Lynch 2016, McKinnon 2020, from the Cannapedia page, REN 2021, and specific NGS from certain publications and NGS data from the genome assembly projects. So the NGS or the variant calling where we use GATK pipeline, and in collaboration with our computing center, we also use a Parabricks pipeline. Parabricks is the same with or follows the GATK algorithm, but it is implemented in GPU, which made this faster, the analysis fast. So, for example, you select the reference and the data set. The data set consists of the cultivar set, which is the list of samples, and the SNP set, which is generated from variant calling. And then you select the region or the locus. And then you can show which SNPs and highlight synonymous and non-synonymous SNPs. Okay, it's processing. Okay. So the result here is in a genotype matrix format. And for this data set, for example, there are 257 varieties, and the region 10 kb long had 673 SNP positions. You can span and look at certain synonymous, non-synonymous. Here you can see the synonymous, non-synonymous, the heterozygous, and also the allele frequencies. So minor allele, major allele. So you can see here which alleles have high allele frequency, minor allele frequencies, and which are rare alleles. And you can download the matrix in other formats, in CSV, tab, link, or flapjack formats. Under development is we are making a haplotype view from this matrix. Actually, it's just an implementation phase. And then the genetic maps. The genetic maps, we loaded sample map from publications. And if we look at one map, for example, the genetic map or the map can be genetic or physical map. And physical map, they name it in linkage groups. And this map also has QTL data. So if we look at one region, okay, so here this is the genetic map, and these are the SNPs, and this is the QTL. You can pan and you see the SNPs in the QTL. You can also compare genetic and physical map. From here, once you already have the genetic map here, it has also you also have to load the physical map. Since there are SNPs, the SNPs are used as physical map. So the linkage group here, LG2, corresponds to the link to the config, this config. And there are 600 SNPs that are common in the map, and they are compared here. So in the left are the genetic maps in Centimorgan, and in the left are is the physical map in base pair coordinate. Okay. So you can color and add more maps to this diagram. And then for Synteny, Synteny is the comparison between different chromosomes or different species, or for different varieties in the same species. And right now, this comparison is based on the alignment of genome of genes. So if there are genes in the same order from one assembly to another, there's a Synteny. So we select the genome, like the reference, and then genome and chromosomes, and then you choose what to compare. So here right now we only have two for demonstration, or that is loaded now. Okay, so here comparison of the CS10 reference with the purple kush query. So these are the regions from CS10 that aligned with the other with the regions in purple kush. And for CS10 with itself, okay, this is the region from this config that align with the other configs. Okay. So a line here means there's a Synteny, and a detailed look at here will show the order of the genes that are considered for the Synteny. Again, since this Synteny uses genes and only CS10 is publicly available, we use the genes generated from our gene prediction for purple kush, and also using for phenola. Okay. In the Phenome, we have the visualization for expression, mRNA and protein expression, phenotype, and biological pathways. Let's see the expression viewer. The expression viewer displays a heat map of the different genes and or proteins in different conditions. And these data are loaded directly from the results from published expression experiments available in GEO, in NCBI. The currently loaded datasets are here in the page. And so there is no meta-analysis where we combine all experiments into one large table. These are loaded individually. And to view the data, you have to enter gene names or gene accession. So if we try an example, so this example displays the experiments, which is the publication where these genes were used. So I named the publications, I named the analysis by the publication title and the author year. So if you select a publication here, it will display the heat map. The heat map consists of the feature, which is, it doesn't show all, it just show the ones that were queried here in the query, in the dialog. And but it shows all samples or conditions where the feature is used, and it shows the detail. If you now some experiments have properties for the samples, and these properties of the samples were loaded directly from NCBI biosample. Some experiments in NCBI don't have detailed description on the biosample, so they're not loaded. But for those with detailed description, they will come up in this selection box here. And then for the phenotypes are also taken from these publications. The phenotypes mostly are molecules or metabolite concentrations. And again, they are loaded per publication. They are not combined into one big table. So to get a distribution or to view the phenotype, they are displayed as trait distribution. So you have to select which experiment or trait you are interested in, Experiment means which publication were they from. Traits means what molecule or trait are you interested in, and then you select the again you have to select you have to select both the experiment and the trait, but it just different order what you select first. Okay, so once you select the publication and the trait, you have a method and then a unit. The note I note here that this module uses the crop ontology form where there's a trait, there's a method, there's a scale, and since we do not have ontology specifically for cannabis yet for these methods, I just use the publication and just the publication name as the method, but ideally this has to be using ontologies. And then once you have the method and units, you generate the chart, and so it displays the distribution of the trait for this experiment. Okay, so that's phenotype viewer. Then the biological pathways, what we use is MapMan. If you're familiar with MapMan, it's a map, a biological pathways map where you can overlay expression data. Here, this is a web version of MapMan. So we embedded this tool here and use the expression data that were loaded in the expression module to color the different pathways. So for example here, if you select which experiment, then you can select the different conditions. Okay. So this is an expression. This is basically an expression viewer, similar to the expression heat map, but organized or layout in terms of biological pathways. So to get the pathways, we have to do a mapping. So we map the CS10 to Arabidopsis genes, or CS10 to E. grandes, or CS10 to Lycopyrrhizicum, which is tomato. Then MapMan has a library of maps with these genes, and we use that maps from cannabis to the map specific to that organism available in MapMan. So that's how we come up with this visualization. So those are basically the main features of the site, specifically the genomics, phenomics features. And there's also a placeholder here we call a virtual gene bank. Its purpose is to have a collection of genetic resources like a gene bank, but since we didn't have a large gene bank, physical gene bank, which holds global data or global seeds. What we have is a virtual. So it's like, if you're familiar with the Genesis platform, it's like a catalog of all gene banks for any species, which physically stays in the gene banks scattered around the world, but the information is available in one site. And for crops, it's available in Genesis, but for cannabis, since it's a new species, or no, it's newly being of interest, it's not available in Genesis. So we wish to make that site or that functionality uh, available here. Okay, so those are the main features of the site, and other features are based on mostly administrative or what's that, information technology related, like commenting on pages and uh, forum, having a chat forum, which can be implemented because these are like standard, or these are modules already available in Drupal, or Drupal based on Drupal, which is a popular content management system. Okay, that's all I have to show you for now, and we welcome questions. Thank you.